Hello, hello, hello. Welcome to the next portion of protein expression, purification, and identification. This time we're talking about protein purification, my favorite. For our learning objectives, you're going to be able to describe the overall protocol for protein purification. You're going to be able to identify scenarios when differential centrifugation is merited. Determine which major type or types of column chromatography would be best for purifying a protein with a given set of characteristics. And finally, you're going to be able to determine when your protein of interest will elute from a column relative to when all the contaminants you don't want will elute. Just to give you a roadmap, we're going to recap protein expression very quickly. Then we're going to move on to lysis, centrifugation. Then we're going to cover the basics of chromatography. This may look a little bit familiar if you watch the protein expression video. So you have bacteria that have been transformed. They have a plasmid that contains your gene that will encode your protein of interest. You pick a colony from your plate, inoculate a small amount of media, and grow it overnight. You use the bacteria from this first inoculation to inoculate a larger volume, and you grow that until you have enough bacteria to induce. You reduce your temperature, you induce with whatever molecule, usually IPTG, but there are lots of others, and you will express anywhere from 4 to 18 hours. After that, you pellet the bacteria and dispose of the media, store the pellet for later use. So we're picking up with lysis next. So you have your pellet, maybe you resuspended it in some kind of lysis buffer, or you just put the whole pellet into the freezer. That's where we're picking up next. You've got your pellet, now we have to do something with it. The first thing you have to do to be able to access your protein of interest is crack those cells open. That's called homogenization or lysis. I usually use the term lysis. It's shorter. I'm lazy sometimes. So you break open the cells to release the cellular contents. And what you get is called a homogenate or lysate. I'll use lysate. Again, it's a shorter word. There are lots of techniques for breaking open your cells. You have, some physical techniques like a homogenizer, which is a plunger pretty much, that fits very snugly into a tube, and you just push and pull on the plunger and you just crack open the cells that way. You can use sonication, which uses sound waves to crack open the cells. You can use chemical reagent lysis, so you pretty much make those cells swell up. You use detergents, poke holes in the cell wall, and then you force all of the contents of the cell out. Sounds pretty forceful and violent when you think about it. And it kind of is, just on a very teeny tiny scale. After you have your lysate, you need to spin your lysate in a centrifuge. That way you're going to be able to isolate your soluble fraction from your insoluble fraction. The soluble fraction has all sorts of things like cytosolic proteins, because remember, the bacteria, it's like a living organism. So it's got its own things going on. It's got its own proteins, it's got nucleic acids, there's ribosomes, all that stuff is going to be in the soluble portion. In the insoluble portion, you're going to have unbroken cells, nuclei. If you're using something other than bacteria, like yeast, you're going to have cell wall or cell membrane, those sorts of things. That's going to be in the pellet, okay? So if you're just spinning your lysate one time, you're going to have a soluble fraction and an insoluble fraction. Chances are the goal of your purification and everything is to have your POI in the soluble fraction. It's the easiest thing to work with, but there are definitely exceptions. Let's say that your POI is associated with an organelle. So maybe you use yeast instead of bacteria, and this protein is associated with mitochondria. What you would want to do is use differential centrifugation. And that, you spin your lysate several times, you're increasing the speed each time to isolate the various organelles. So maybe the first spin, you don't want to keep the lysate because what you want is in the pellet. Then you resuspend your pellet in some kind of buffer and spin again. And then maybe you have what you want. So why go through all the trouble? If you need to isolate a specific organelle, or maybe you need the membrane from something, then you may want to use differential centrifugation so that you can remove more contaminants and enrich for your lysate. Enrich the lysate for your POI. Otherwise, don't go through the trouble. So when it comes to choosing what to keep and what to toss for your purification, otherwise, who's voted off the island? Many proteins are soluble, or you can make them soluble so that when you do your centrifugation, you're keeping the soluble portion. But there are other proteins that may be localized to different organelles or the cell membrane. They're associated strongly with the cell membrane. So you might actually need to keep the insoluble pellet and move on from there. So the short answer is it depends. The caveat is you should always keep everything when you're doing a purification. And that's just a practical tip. Until you are very comfortable and you've done this procedure many times with great results, do not get rid of anything until you are done and you have your protein and you've confirmed its identity. So just hoard everything until the end. So this brings us to chromatography. We have to have some way of separating our POI from everything else. I'm going to cover up half of this slide. It's a little bit intimidating. So chromatography takes advantage of the fact that if you have a mixture of different compounds, they're going to distribute themselves in specific ways based on the properties that they have. So it's kind of like a like dissolves like sort of thing. If you've got a lot of polar molecules and you're using chromatography where you have resin that's polar, those polar molecules are going to want to hang out more with the resin and take a longer time to elute from whatever column you have or to move through whatever stationary phase you have. And I'll talk about what the stationary phase is in a minute. But if you have something that's nonpolar in a polar environment, it's not really going to want to stick around. So it's going to try to move away from the polar resin or whatever your stationary phase is, whatever thing you're using to separate the different compounds in your solution. So I've used the word stationary phase. There's also a mobile phase. So let's talk about those two words. The stationary phase is the substance that selectively slows the flow of your sample and causes the components of the sample to separate. So in our case, we're going to be talking about column chromatography, and the stationary phase is going to be a column that's packed with some kind of resin. And that resin has some kind of ligands or something special about it that will hopefully retain your protein of interest and allow you to wash away all of the contaminants you don't want to keep. The mobile phase is pretty much the mixture to be separated. In this case, it's our lysate. It contains our POI. You pour your mobile phase onto your stationary phase, and then it flows through. So I will be using terms like column chromatography, liquid chromatography, and LC. They are all the same thing. You have a column packed with resin, and you apply your lysate, your mobile phase, to the column, and then you collect fractions. Wouldn't be complete without a picture. You knew it was too long. We didn't have any pictures. It was a lot of words. Nah, we got to have a figure. Okay. So this is my poorly drawn column. It's actually not that bad. I'm a little bit proud of it. So you have a column. This pink here, this is the stationary phase. So whatever resin you're using, this is where it is. You take your lysate, you apply it to the column, and it will slowly trickle through, and you collect the flow through. So everything that didn't bind to the column, you're going to collect all that. Keep it, but hopefully you don't need it. There are some cases where, for whatever reason, your protein of interest didn't stick, or maybe all of it didn't stick and you need to reapply. So you keep that flow through, and in some cases, practically speaking, you will reapply that to the column before you move on. Over time, and with whatever washing and stuff you do to the column, sometimes you need to incubate before you start washing so that your POI has a chance to bind with the resin. You'll have some kind of separation of the different molecules in your sample. Molecule A is still pretty close to the top of the resin, B is sort of in the middle, and C is trying to bust out. And that's because each molecule has different levels of interaction with the column contents. So the faster you move through the column, the fewer interactions you have. The longer it takes for you to mosey on through the column, the higher your retention time is. So in this case, molecule A is going to elute last because it's taking its sweet old time. It's saying hi to everybody. Hey, how you doing? Carl, how's the wife? Like molecule A is chilling, okay? Molecule B is, the cordial nod. Hey, how you going? How's it going? How's it going? But they're not really stopping to say hello and really see how people are doing. I don't like it when people do that. Anyway, molecule C is like, look, I have social anxiety. We should be socially distancing ourselves. I'm getting out of here. So it will elute first. So the take-home point here is the tighter the interaction between your protein of interest and the stationary phase, the higher the retention time. So that means that you'll be able to wash your column a little bit more stringently and get all the junk off that you don't want. There are three major categories of LC that are used for protein purification. This is absolutely from a biochemist's point of view. Chemists also use LC, but when they're doing their small molecule stuff, I don't know what they'd be doing. No, that's actually not true. But these three different types are very heavily used when purifying proteins. The first category that we're going to talk about is size exclusion. So size exclusion separates based on size. Pretty logical, self-explanatory. And your stationary phase, so the resin that does the separating, is just composed of cross-linked gel particles that form pores of various sizes. So this was my pore drawing of the purple and the blue little dots. Those are the gel particles of various sizes. And this is a channel right here for protein to flow through, channel right here for protein to flow through. Your protein has to be within the molecular weight cutoff range. So the molecular weight cutoff, the MWCO, that dictates the lowest molecular weight that's retained by the column. So anything bigger than, say, if your column has a 20 kilodalton molecular cutoff, that means that any protein this size or larger, you can separate. The way that it works is that the large proteins bypass the pores. So they're not even bothered. They're like, okay, yeah, I see this. It's a little bit of a jungle, too much of a tight squeeze for me. I'm just going to go around. That means that they elute first. The smaller proteins are like, ooh, I like this. It's a jungle gym. Let's go. So they will interact with the cross-linked gel particles and it will slow them down. The smallest protein in your mixture will elute last. Size exclusion is best used as a final step in purification because you can actually use it to remove salt and metal and things like that from your protein. So if your protein is sensitive to metals or salts or high salt, then you can use it to do that. Also, if you have like a contaminant that's a protein that is a distinctly different size from your POI, you can also use size exclusion to get rid of that contaminant. So it's great as a final step. It takes forever though. The columns are sometimes really long, like we're talking, 2 or 3 feet long. So it can take a while, but it's really nice. The next category we're going to talk about is affinity chromatography. Now, this is a really big, broad category. So we're just going to kind of take it step by step, and I'll give you some examples. So overall, affinity chromatography separates proteins based on their affinity to a certain type of ligand that's bound to the resin. So you'll have some kind of agarose or something like that, and then you'll have a protein or some other kind of ligand for your stationary phase. So the stationary phase varies greatly. A very common example is agarose that's covalently bound to protein A. Protein A is, it binds really tightly to IgG type antibodies. So if you have something where you can use that to your advantage, you can have some kind of a tag on your protein that enables it to interact with protein A, and then all the other stuff will flow through. You can also use immobilized metal affinity chromatography, or IMAC, and that's where you use a metal like nickel or cobalt. The nickel is a pretty blue color. The cobalt is like a pink color. And your protein will interact with the metal, and it'll hang on to the metal, and everything else will flow through. That's not quite as specific as, say, using protein A, but it still works quite well. So the way this works, we're going to cover up some of this. So your POI has a high level of interaction with whatever ligand is bound to your gel matrix. So whatever agarose plus protein or metal or something like that. You use a high salt buffer to wash the contaminants away. And then you elute with either very high salt buffer or with a ligand that has greater binding affinity than your protein of interest. So in the case of protein A, you're going to use a protein ligand that will replace your POI. Instead of your POI being bound, it will be the ligand, and it'll flow through the column. The high salt will interrupt interactions. So very high salt concentrations will force your protein of interest off the column. Oftentimes, with the affinity method, your protein of interest has some kind of N or C terminal tag that binds specifically with high affinity to a certain ligand. So that's something that we're not really going to talk about in detail here, but practically speaking, using tags on either end of the protein is a very effective way of isolating a protein because you can manipulate it even more. So affinity chromatography is very powerful. There's lots of different ways that you can do it, and it's highly advisable that you use affinity chromatography as part of your purification protocol. I don't feel as high energy this morning, y'all. Must be the rain or something. It's raining a lot. Let's try to get that energy up. Woohoo! Ion exchange, yeah. No, that didn't work. All right. So the final topic for chromatography is ion exchange, and as the name implies, this separates proteins based on charge. So you have a stationary phase with a gel-like resin. You've heard that before. And there's a charged molecule that's cross-linked to it. So you can have a cation exchanger that will retain positively charged proteins because the resin is negatively charged. So the cation part talks about what it will retain. So it retains cations or positively charged ions. Anion exchanger retains negatively charged proteins. Your resin is positively charged. So again, the anion part is for what it retains, the negatively charged particles. So here, you run your lysate over the column, you wash it, and then you use increasingly higher salt concentrations, or you use a pH change with your buffer to elute your protein of interest. So the elution is kind of similar to the affinity columns, where you're using salt or something to replace your POI. And you have to remember that proteins are made of amino acids, and those amino acids have functional groups that can be affected by pH. So when you're making your buffer, you have to take into account that your pH of your buffer can make your POI positively charged or negatively charged. So that really depends on what the PI of your protein is. And if you remember from when you talked about proteins in class, hopefully, everything has an isoelectric point. So the amino acids do, and so does your protein of interest. There are websites where you can put in your amino acid sequence and it will predict your PI. That's very valuable information for when you're making your buffers. When you're making your buffer, you have to remember that if it's anything, so here's our PI, if it's anything below, then your protein is going to be positively charged. So let's say that your PI is 6. So we're talking about what pH is your protein have a neutral charge. Anything below 6 for a pH, that protein is going to be positively charged. Anything above 6 for the pH is going to be negatively charged. So you can use that to your advantage. Ion exchange is best used in like a first step. It's not super specific. DNA and RNA, those are charged molecules, so they will absolutely interact with an anion exchanger because they're usually negatively charged. Cytosolic proteins from whatever bacteria or yeast, whatever system you use to express your protein, they're absolutely going to be affected by the pH of your buffer. So some of those proteins will be made positively charged, some of them will be negatively charged, and some of them will interact with your ion exchanger. So take it with a grain of salt. It's a helpful tool, but I would not use it as a final step because you have way too many impurities. So here's an example to give you a better idea of how ion exchange works. So let's say that you have DEAE cellulose, which is an anion exchanger, so it's going to be positively charged. Don't worry about what this stands for. This is the molecule that's cross-linked to cellulose to make this resin. Oh, I'm giving you guys the answers. It's all right, I was going to give them anyway. So you're separating a mixture of amino acids. You have aspartate, histidine, and valine. What order would they elute from the column? So aspartate has a carboxylic acid as a functional group. So that's its R, okay? That's going to be negatively charged. Histidine, it has an imidazole as a functional group. It's positively charged. Valine has a nonpolar R group. It's a hydrocarbon. So that's neutral. If we have something that is positively charged for our resin, that means something that's negatively charged is absolutely going to love hanging out with it. So it's going to elute first. It's going to elute last. It's still early, y'all. The imidazole group with the histidine, positively charged, positive and positive, those are going to repel. So it's going to elute first. And the valine is kind of in the middle. Remember that all amino acids have a carboxylic acid and an amine. That's part of the backbone. So we're not really taking that into consideration. So the valine does have some functional groups that will interact with the DEAE cellulose, but the side chain doesn't really contribute to those interactions. So it's going to come off kind of in between the two. Nothing to hold it there strongly, but nothing to really, kick it off really soon. What I want to end with is talking about the technology that we have for completing chromatography. So liquid chromatography takes forever by gravity. You're setting it up on the benchtop. Maybe you're in the cold room, wearing a coat because it's cold. It takes a long time waiting for everything to drip through slowly, 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 especially with the lysate. The lysate is kind of viscous sometimes, and it just takes a long time. Nobody wants to be there forever. If you're a graduate student, you want to leave. Eventually, you want to go home and eat, go to sleep. So using HPLC and FPLC, those are fast automated options for protein purification and small molecule purification. So HPLC is high performance liquid chromatography and FPLC is fast protein liquid chromatography. I have done a lot of FPLC for protein purification. I've used HPLC for separation with proteins, but not necessarily for purification. FPLC is wonderful. You set up a program on a computer, you load up your column, you put your buffers in and everything. This Mamma Jamma will do all the things for you. It controls the equipment and it collects everything, all your fractions, and some of them will also read the absorbance of whatever is coming off the column and tell you, okay, there's a peak here, so using 280 nanometers, that's usually what you use to detect protein. You can look at the spectrum, see where you have a big peak, and you can use, samples of those tubes to identify your protein. So it's great. I would not fly without it. So if you are thinking about grad school and doing anything with protein purification, make sure your lab has an FPLC or you have access to one, or else it's going to be struggle bus. Okay. So apply your knowledge. We talked about a lot of things today. So you're definitely going to want to give this a try. Make sure that you have a grasp on how to put together a protein purification protocol and that you understand the basic principles of how the three major types of chromatography work. So in this premise, you have to develop a protocol to purify a small protein, and you're given the molecular weight and the PI of the protein. So the grad student that passed off this project had a high molecular weight contaminant present in their sample. So when you're designing this protocol, you need to use size exclusion and ion exchange to isolate the small protein. So just keep in mind all the details for how to do this. Don't forget to include lysis and centrifugation as part of your protocol because those are important as well. This protein is, it's a small protein, it's soluble. Okay. Please include what pH your buffer is for the ion exchange. And then also include when your protein will elute from size exclusion. You don't have to say, it will elute at fraction 52, but tell me, will it elute, early on, in the middle, at the end? And then let's flip it a little bit. Let's say that your small protein was, instead of being soluble, it is strongly associated with the cell membrane. How would this affect your centrifugation step and just what fractions you would carry forward? So that's it. Give it a try and see how you do. If you need some help, you know how to get to me. Leave a comment, email, all that good stuff. Have a good one.